الرحمن الرحيم. إني الأستاذ المساعد الدكتور احسان فليح حسين من جامعة بابل كلية طب حمورابي. Our lecture today is general concepts of antibiotic. Antibiotic are a chemical substance produced by living organisms, generally a microorganism that kill or inhibit the growth of other microorganisms. Antibiotic re-evaluations medicines in 20th century. This development by Alexander Fleming discovered penicillin. Now today is antibiotic that are used in the treatment of a wide range of disease. This scientist is discovering this disease by chance when the study of pathogenic microorganisms, especially bacteria, on this disc. He noted this disc is contaminated with fungus and noted this clear zone. This means this called the pathogenic bacteria. He studied what's the material that causes the kill or inhibit the growth of pathogenic bacteria now and today as antibiotic. There's several types of antibiotic. We noted that causes inhibit the growth of pathogenic microorganism on the disc by noted the clear zone. This means sensitive to antibiotic, while this antibiotic is resistance. The antibiotic can be divided into two categories, antibiotic and antimicrobial. What are the differences between these two means? Antibiotic is term used to describe a natural compound that is used to treat bacteria infections inside the body, while antimicrobial is a greater comparison from any natural or artificial compound that inhibit or kill multitude of microorganisms, bacteria, fungi, viruses, protozoa, and other. This means antimicrobial when have activity against fungi called antifungal or antiviral or antiprotozoal and other. This means a wide range activity by compared to the antibiotic. Antibiotic can be taken in different ways: orally, typically through the injection, intramesals or intravasally, non-IV or IM. What mean the differences between this method where take antibiotic? This depended on several, or according to several causes. Some antibiotic can cause toxic when obtained orally, or can break down by some acids in the stomach. Therefore, we must take this antibiotic to treat it of several infections by bacteria. Therefore, we taking according to injection. Some antibiotic can cause toxic to the human when taking in IM or IV or also orally, but can use this antibiotic topically as cream, spray, ointment, and other. This figure shows several types of antibiotic. These capsules, when taken orally, we noted this capsule as capsule because this breakdown in the stomach can cause toxicity to the human. Therefore, cover it because this causes some injuries to the mouth and other tissues. Here, injections, topically, and other type of antibiotic, spray, drops, ointment. How do antibiotic work? The antibiotic can work in one of two ways: the bactericidal antibiotic and the bacteriostatic antibiotic. The bactericidal, such as penicillin and other antibiotics, act like kill the microorganism. This means the microorganism cannot grow in latent time. The bactericidal, this causes inhibit or stop growth or multiply the microorganism. And this means this causes to help to immune system and cause remove infected bacteria. Antibacterial spectrum. Antibacterial spectrum is the mean that range of microorganisms it can kill or inhibit. Antibiotic can be divided into three types or three categories: a broad spectrum antibiotic, extended spectrum antibiotic, narrow spectrum antibiotic. A broad spectrum antibiotic can kill or inhibit a wide range of microorganisms. Example, cephalosporin. Extended spectrum of antibiotic can kill or inhibit gram-positive bacteria and some gram-negative bacteria. What's mean gram-positive and gram-negative? These two types of bacteria, the scientists classify this bacteria according to the structure of cell membrane, cell wall, and other compound. The antibiotic that have activity against pathogenic gram-positive bacteria, this causes breakdown of the peptidoglycan, the main compound of structure of the cell wall. Narrow spectrum of antibiotic can only kill or inhibit a limited species of bacteria, example, vancomycin. Mechanism of antibiotic action. The antibiotic have 5 main mechanisms of action by which can kill or inhibit the growth of pathogenic microorganisms. Number 1, inhibition of cell wall synthesis, inhibition of protein synthesis, alteration of cell wall membrane, inhibition of nucleic acid synthesis, and anti-metabolite activity. This figure shows the main mechanism action of antibiotic against pathogenic bacteria and give some example of antibiotic that act by this mechanism. Cell wall inhibition. The main function of cell wall is protect the microorganism from inappropriate condition. Therefore, when a breakdown of the cell wall, this causes bacteria. Some beta-lactam drugs, some beta-lactam drugs that kill cell wall or that kill bacteria by a breakdown of the cell wall, penicillin, cephalosporin, and other. Vancomycin, pastoracin also act by killing the cell wall. Cell membrane. The main function of cell membrane is control of movement across outside and inside of the microorganism, such as nutrition, fluids, and other. When the breakdown of the cell membrane, this called kill the pathogenic microorganism, such as polymyxin. Protein synthesis. We all know all living organisms and microorganisms need protein for continuous life. When stop the synthesis of protein, this causes kill microorganisms. Some antibiotics act on 15 subunits and other 13 subunits, tetracycline and aminoglycoside, that act on the 13 subunit, while macrolide, clindamycin, and other act on 15 subunit. This gives the selective between activity against human or eukaryotic cells and prokaryotic cells. We know the large subunit is consists of 16 subunits, while the small subunit consists of 14 subunits. This give idea about this antibiotic can kill the pathogenic bacteria and not effective on any human or eukaryotic cells. In nucleic acid synthesis, some antibiotic can cause a breakdown or breakdown the nucleic acid. They act exactly on the enzymes that made the nucleic acid, such as rifampin that break down the RNA polymerase and the guanylines that causes the breakdown DNA galactose. Mechanism of antibiotic resistance. The bacteria can cause resistance against the antibiotic by several mechanisms. The main mechanisms of resistance can limited uptake of antibiotic, limited uptake of an antibiotic, modification of an antibiotic target, inactivation of an antibiotic, active efflux of an antibiotic. This figure shows the main mechanism by which the pathogenic microorganism can resist the antibiotic. We noted impermeability modified of cell wall protein. As we know before this, the antibiotic can cause a breakdown the cell wall. Some microorganisms can resist the antibiotic by change or modify the impermeability of the cell wall. Also, inactivation of antibiotic. Note this antibiotic can cause modification, added a group or remove a group by several enzymes. This causes change the main structures of antibiotic, result in inactivation of antibiotic against bacteria. Some microorganisms can take antibiotic inside and they cause shorting bulb to halt. By this way, the antibiotic cannot reach to the target, therefore not causes any effect against this bacteria. Modification of target. We all know when any antibiotic have special targets such as cell wall, cell membrane, protein synthesis, ribosomes, especially alteration of modified. By alteration the antibiotic target, the antibiotic cannot react or join with the specific target. By this way, the antibiotic cannot give any activity against this bacteria, and bacteria can cause resistance to the antibiotic and continuous ingrowth and causes infection to the human and other animals. This figure shows the antibiotic resistance may be caused by chromosomal gene within the genetic system of microorganism that causes resistance to some antibiotic, or by transformation through the enter of plasmid. Some plasmid is circle DNA that causes some gene. Some of this gene can cause resistance to antibiotic. Also, we know that antibiotic break down by enzyme that cause degradation of antibiotic, afflex bar, antibiotic resistance gene that present with the plasmid causes the several type of resistance. Antibiotic not enter to the cell. Some pathological bacteria causes hidden antibiotic by clotted it. Antibiotic alteration enzyme, alteration antibiotic target. How we know the microorganism, especially pathological microorganism, can resist the antibiotic? This figure shows this. Note clear zone. This means the antibiotic is sensitive. Not enough antibiotic is resistance. This continues with the time, increase the resistance by misuse of antibiotic. Side effect of antibiotic. Commonly, the antibiotic or uses of antibiotic cause several types of side effect. Commonly causes side effect: diarrhea, nausea, vomiting, rash, upset stomach. The prolonged use of certain antibiotic can cause fungal infection of mouth, digestive tract, and vagina. Less common side effect of antibiotic: formation of kidney stone when taking sulfonamide, abnormal blood clotting when taking some cephalosporin, sensitive to the light when taking tetracycline, a blood disorder when taking trimethoprim, defense of taking erythromycin and aminoglycoside. Thank you very much.